The overall goal of the following experiment is to quantify the changes in confined phase behavior and transport properties of attractive colloidal suspensions using confocal microscopy. This is achieved by formulating colloid-polymer mixtures to model attractive colloidal suspensions. The mixtures are imaged and confined in thin wedge cells to show how confinement modifies the structure, dynamics, and phase behavior of these mixtures. Next, mixtures are imaged as they flow through microchannels to determine the effect of the strength of attraction between the colloids on the confined transport properties of the mixtures. Automated image analysis and particle tracking algorithms can show how quiescent confinement leads to solidification, and image correlation algorithms can show how increasing strength of attraction leads to increasingly non-Newtonian flow behavior.
The main advantage of this technique over other methods like electron microscopy and dynamic light scattering is that it allows direct access to single particle trajectories from which dynamic and structural metrics can be computed for direct comparison to simulations.
This standard method can help answer key questions in the field of soft condensed matter physics, such as how suspensions self-assemble in the presence of interparticle interactions, or how solid-like suspensions yield under an applied force or stress.
For this protocol, have prepared PMMA particles that are labeled with a dye. Consult the linked reference material in the text protocol for standard preparations. Make a 3:1 mixture by weight of bromocyclohexane in decahydronaphthalene. Next, add tetrabutyl ammonium, an organic